Hello, we are continuing to highlight the participants of the virtual research poster competition. Joining me today is Nick Juhasz. Hi, Nick. How are you?
Hey, Kyle, I'm doing well. How are you?
Good. So tell us a little bit about yourself.
Yeah, so I'm actually a recent graduate of NC State. As of last December, I got my degree in genetics, and now I'm continuing to work in the vector-borne disease diagnostic lab.
Great. Who else is in that lab?
So right now I work under Dr. Carullo, uh, which is my main mentor for this project.
And tell us about the research you've been doing.
Yeah, so our goal, I work with Dr. Carullo and some collaborators from Duke to assist in distinguishing between some Leishmania species that are common in Peru. And Leishmania is a vector-borne pathogen that's spread by the sandfly that can cause pretty painful lesions on the skin. And specifically the region that we work in, the Department of San Martin, has reported the most cases of cutaneous leishmaniasis in Peru over the past 15 years, as well as a lot of mucocutaneous leishmaniasis, which is a lot more severe and difficult to treat. And this region supports multiple species of Leishmania, probably around more than 5, due to its various ecoregions. So we're trying to sort of distinguish which species are most prevalent in forms of the disease and as well as treatments as well. And to make matters a little bit more difficult, there's been a possible identification of a hybrid species. And what that kind of means is that Leishmania has 2 separate genomes, nuclear and a mitochondrial. And so whenever we get our samples, we test for both genomes, and we've noticed a species discordance. And that's just one species on like the nuclear genome and another species on the mitochondrial. So far we've received 52 samples from the health clinic in San Martin, and of those 28 have tested positive through our PCRs. 11 of them have been specific to one species of Leishmania, and then 17 of them have been consistent with this mitonuclear discordant strain. And one interesting thing that we did notice was that these discordant strains were a lot more prevalent among all of the infected men. And we're not exactly sure what that means yet, but we also don't have all the clinical data from the people yet. But once we get that, we can probably start to draw a little bit stronger conclusions on what that means. And so we think that these results really highlight a need for more investigation towards the significance of this mitonuclear discordant strain of Leishmania. These discordant strains and species may add to the difficulty in identifying species and as well as treating the people in the region.
Interesting. More to research.
Yes, always.
Well, thank you for sharing, Nick.
Thanks for your time.